Hello, everyone. My name is Hailiang Huang. I'm a statistical geneticist at the Broad Institute. I'm very happy today to do this GWAS tutorial with you. So the first thing, let's clone the GitHub I have created for you, the GitHub repository that I have created for you with the data and tutorial materials. Please type this in your command line, in the terminal, the git clone and the location of the repository under my first and last name and GWAS-tutorial. It will take a while, but it should be very fast. All right, this is done. Let's get into the location, the folder you just cloned, and have a look at all the files. I would recommend you to open the GWAS_tutorial and in your text editor so that you can just simply copy and paste everything from the text editor to your terminal. All right, I have opened this in one of my text editors, and just notice the few information here, so first, where you can download this GitHub repository. In case you have any question, feel free to contact me. And for this tutorial, you have to install the Pinlink tool, which can be retrieved here, and R, which can be downloaded from the R project website. And also, we use the QQman package in R, so please have it installed in your R program. All right, let's get started. So first, let's take a look at the data. So this is one of the two data files. It has individual level genotype. If you use the head command, you will see the first few rows in that file. The first two columns are the individual ID and the family ID. The next two columns are the IDs for the parents of that individual and the sex and the diagnosis. After the first 6 columns, the rest of the columns have the genotype of that individual, which you can easily tell. And in case that genotype is missing, we have 0 to indicate the missing genotypes. And the other file is called a map file. It basically has a list of SNPs, and each row here corresponds to the every 2 columns here, the map 1-on-1, so that Pinlink will know which SNP this data set is. All right, so the first thing we typically do after we see this data set is to convert this to binary. What you saw is very convenient for you to take a look at what the data is, but there's a format called binary format that is more efficient in terms of the storage and analysis. All you need to do is to indicate your input file, which can be done by the -- file, and you can use the command --makebed to convert this text file to binary format, and then indicate where you want this binary format to be written to in the --out command. All right, this is done. And if you compare the size of the file before and after the conversion to the binary file, here is how you can do that. So you can see that before conversion, you have 77 megabyte for the file with the genotypes, and after the conversion, you only have 4.7 megabyte. So the conversion saves you more than 10 times space. All right, so let's start some QC. The first thing we want to do is to remove the individuals that has a high missing rate. You can do that by using the Pinlink --missing command. The --b file simply tells Pinlink where the input files should be. The --out tells Pinlink where Pinlink has to write the output files to. So you use the --missing command to calculate the missingness, and here is what the output file looks like. It basically has a list of individuals with their missing rate. And here, the out command filtering on the missing rate in the 6th column, you can tell we have 2 individuals with more than 2% missing rate. A high missing rate typically indicates problems in the sample quality. So it's not a bad idea to remove the samples with high missing rate. All right, So the next we want to do is to remove samples with high heterozygosity rate, and this can be done by calculating the heterozygosity rate using the --head command. And if you take a look, this is the output from that calculation. It's basically a list of individuals with their heterozygosity rate. I have prepared a R script for you to put together individuals that fail either the missing weight test or the heterozygosity rate test. And for considering the time, I'm not going to go into the details of that R script, but you can just call that R script by doing this. And that script will write a list of individuals with extremely high missing rate or with heterozygosity rate that are either too high or too low to this fire. And all you need to do is to use the --remove command to remove that list of individuals and tell P-Link that you want to write the new data file to this file. All right. So we have removed individuals that have a high missing rate, but we also have to do that for the SNPs, to remove the SNPs that have a high missing genotype rate. We can do that by leveraging the other output file from the --missing P-Link, and that is a iMissing file, which has a list of the SNPs with their missing rate. And we decide to use 5% as a threshold here. We write all the SNPs we want to exclude in this file. And we use the P-Link --exclude to remove such SNPs. So P-Link --remove removes individuals, and the P-Link --exclude exclude the SNPs. All right, this is done. We now have the second iteration of QC with the SNPs with high missing rate removed. The next we want to do is we want to find SNPs with with differential missing rate. The missing rate is different in cases versus in controls. We do so by using the --test -missing here, and take a look at what is inside. You have a list of the SNPs with their missing rate in cases as affected and controls as unaffected. In this case, we don't want to use the p-value because the sample size is too small for the p-value to be helpful. We simply want to remove the SNPs with 5% difference between cases and controls. And this can be done by subtracting the difference, for the difference between the missing rate in cases versus controls, and using 5% as a threshold. We write everything we want to exclude here, and we will remove that list of the SNPs here. All right. The last thing we want to do on the SNPs is the Hardy-Weinberger equilibrium test. And this can be done by using the --Hardy command. This is a list of SNPs with their Hardy-Weinberger p-value. And we want to only use the Hardy-Weinberger test for the unaffected samples. And we want to use the p-value of 10⁻⁶ as a cutoff. We filter for such SNPs, and we use P-Link to remove those SNPs. All right, now you have a data set that are relatively clean for individuals and for SNPs. What's next? The next is to remove individuals that are related to each other. But to do that, we need to create a data set that is independent in terms of the genetic variance. We can do so by using the --, independent pairwise. And these are a bunch of parameters going to the LD pruning process, including the window size, the step of the window that is moving, and also the threshold you want to use for considering two variants that are in LD with each other. So after you run this command, you will have a list of SNPs that P-Link would recommend you to keep as independent SNPs. This is in the file called -prune-in. All you need to do is to generate a new data set with only this variance by using the -- extract command that will give you a independent set of markers and in a new p-link binary format. And then you want to use the -- genome file to calculate the relatedness between samples and take a look at what is in that output file, which is just a list of pairs of individuals with their relatedness. Typically, a relatedness of pi hat greater than 0.2 indicates something concerning. We want to capture that pairs using this command, write them to this file, and we use the p-link -- remove as we did before to remove such individuals. All right, so let's take a look at how many variants and how many individuals we have removed. It seems that the data is pretty good. Before QC, we had over 100K, 100 individual SNPs and 200 subjects. After QC, we got rid of less than 1,000 variants and 8 variants. All right, so now let's take a look how well we did in the QC. We use the word count -l to count how many variants we have before and after QC and how many individuals we have before and after QC. So it looks like the data was pretty good. The QC removed roughly 1,000 variants, which is very normal. We started with a little over 100,000 and we ended with 99.7 thousand, which is not bad. We removed 8 individuals from the 200 to 192, which is not bad as well. All right, so far so good. So what next? The next is to generate the principal components so that we can include them as covariates to control the population structure. So this is how you do that. You need to create a new independent data set, independent in terms of the variance, and you want to generate that based on the latest file you have created, removing the related individuals. You do that similarly as you did before, using the -- independent pairwise command. Then you create a new file, the file that only has the independent variance, like this. And then you create the principal component plot. You perform the principal component analysis using the -- PCA command. All right. This is output from that command, the dot eigenvalue and dot eigenvector. The dot eigenvector has what you need for the downstream analysis. So let's take a look. It has a list of individuals with principal components, a lot of them. So what you do next is to perform your GWAS with these principal components. And this can be done using the -- logistic, because this is a case control study. You want to use the logistic regression. The height covar basically asks p-link not to write the coefficient for the covariance, because you, for this study, you only care about the variance, the coefficient for the variance, not the covariant. This is to tell the p-link where your covariant is, which file has a covariance, and this is to tell p-link how many components, how many principal components you should include as the covariance. And here we do 1 to 10. All right, let's do it. All right, it's done. So the .logistic file has a list of all the variants with their odds ratio, standard error, the confidence interval, and the p-value. This is exactly what you need from a GWAS. And we could use this little script I wrote in R to do some visualization. That script also calculates the genomic inflation factor, which is only 1.06. Looks like the genome has been very well controlled. And let's look at the 2 figures it has generated. The Manhattan plot, let's open it, and the QQ plot. All right, so this is a Manhattan plot. This is a QQ plot. You can tell that the genome the genome has been well controlled. You don't really have an inflation, but unfortunately for this particular GWAS study, we don't have a genome-wide significant signal for now. We do have, one signal surpassing the suggestive significance threshold, suggesting that we may need more samples for this GWAS to identify the significant locus. All right, that is a very quick tutorial, and I hope it's helpful for your research. Thank you for your attention.